Namaste students, I am JV Dr. Aziz Mohd Khan from Department of Food and Biotechnology, Faculty of Agriculture and Veterinary Science, Jyoti Vidyapeeth Women's University, Jaipur. Today I am going to discuss about the topic lectin pathway of the complement system. Lectin pathway is a pathway which is third pathway after the classical pathway, alternate pathway and the lectin pathway. The lectin pathway is a pathway which is related to the lectin metabolic pathway, and the classical pathway is the basic, which is the original pathway. And when the pathway is not followed, then it goes to the alternate pathway. And when there are some special lectin molecules are involved, then the pathway is known as the lectin pathway. So in this video, I'm going to discuss about the lectin pathway and the mode, how the lectin pathway is conducted, what is the structure of the lectin pathway, protein molecules, and which proteins are involved in the lectin pathway. Lectin pathway is independent of antibodies, so there are no antibodies are involved. Okay. And in the classical pathway and the alternate pathway, there were antibodies which were involved. But in lectin pathway, no antibodies are involved. Lectins are proteins that bind to the specific target molecules, classical binding protein targets. So in this pathway, lectin is the main protein. Lectin, this pathway, lectin binding pathway is activated by mannose binding lectin molecule. Okay. This is a lectin. Mannose binding lectin is a lectin that bind to the mannose residues on the microorganism. Mannose is a sugar molecule, carbohydrate molecule, and the mannose binding lectin will bind to this mannose region of certain microorganism. Mannose binding lectin is similar to C1q in the structure of we have already discussed in the classical pathway that is like that, and reasons C1q, and this is q and this may be r. Okay. Similar structure is observed in the MBA, mannose binding lectin pathway. So what is the structure and what is the activation pathway? First, we discuss about the structure of the. So this is the lectin amyl molecule, and this mannose binding lectin molecule has two molecule, amyl bound molecule, which are similar to C1R and C1S. The first one is MSP1 and MSP2. MSP1 and MSP2. So it is like C1Q, and the two molecules are like C1R and C1S, such as C1 MSP1 and MSP2. What is the full form? MBL associated serine proteases. So this is the MBL and associated serine proteases 1 and MBL associated serine protease 2. Okay. So this MSP will bind to the MBL. So how the activation of lectin pathway occurs? The first step is the binding of the mannose binding lectin to the mannose residues on the surface of the microorganisms. First, the microbes will bind, this MBL will bound to the MBL, mannose residue on the surface of the microbes, and to the MBL bound to the microbes, MBL association, these MSP1 and MSP2 will bound. MSP1 and MSP2, as we have already discussed, that they are similar to the C1S and C1R. This protein complex will then cleave to the C4 and C2. Okay, this is C4. The binding of MSP1 and MSP2 will cleave the C4 into C4A and C4B. The C4A will be removed and C4B will bind to the membrane surface. After that, C2 will be MSP2, activated MSP, then also cleave the C2 region. C2 will be cleaved into C2A and C2B. C2A will be bind to the membrane and C2A will bind to the C4B, but C2B will be cleaved out, removed out. After that, C3 will come and it will be cleaved into C3A and C3B. The C3B will come, okay, it will come and it will bind to the C3, C4B, C2A, making a complex into C4B, C2A, C3B complex. After that, the C5 will come and it will bind, and after that C6 will come and bind, and after that C7, C8 and C9 will come and they will bind to the surface of the uh, uh, membrane, and so much molecule of the C1, C2, C3, C4, C5, C6, C7, C8, C9 will bind on the surface of the microorganisms at different regions. And finally, the C9 region will make a membrane-attack complex, and after that, it will make very different number, a lot of pores on the surface of the microorganisms on different regions. Okay, when it will make the pore on the surface, then these pores will cause the destruction of the bacterial cell. Okay, so what is difference between the classical pathway and the lectin pathway? In the classical pathway, the starting is different. In the lectin pathway, the starting is different. Same type of proteins are involved, but in the classical pathway, the pathway started from binding of antigen and antibody complex, and in the alternate pathway, the pathway starts with the binding of a mannose binding lectin, and it will identify the mannose residues on the surface of the microorganism. If they will identify the mannose, the pathway will start and it will be activated, and the first MBL will bind. After that, MSP1 will bind. After that, MSP2 will bind, which will lead to the binding of the C2A, C4B, C4 and C2, which will lead to the binding of the C3, C5, C6, C7, C8 and C9, which will finally result in the destruction of the cell, bacterial cell or pathogenic cell. So how students will get in touch with this topic and what they will learn? The students will be able to understand the lectin pathway, what is lectin pathway, and the modes of the lectin pathway, the proteins which are involved in the lectin pathway, the structure of the proteins which are involved in the lectin pathway, the complete pathway, how the lectin pathway is different from the classical pathway, and how the lectin pathway starts. which molecule the lectin pathway identify. It identifies the mannose residue on the surface of the microorganism, and also it, the students will be able to understand uh, some diagrammatic representation using this lecture. So, which kind of questions can arise from this lectin pathway? The first question, that what is the lectin pathway and how the lectin pathway starts. The second thing, what are the modes of the lectin pathway? Which proteins are enrolled in the lectin pathway, and how the lectin pathway protein molecules are activated? How these proteins will activate some another proteins, okay, and how the complete lectin pathway will lead to the destruction of the bacterial cell. So this is all about the lectin pathway. So my lecture is over here. Thank you very much.